We're going to move on to the audience Q&A. We have quite a few, and as promised, I want to try to get through as many as we can. So this first one is going to be addressed to both of you. Mike, we'll start with you. Can you specifically address how changing from a lab-based adherent protocol to suspension scalable protocol improved a cell product? Also, what challenges did they meet while performing a process change?
I don't know if it would necessarily improve a product. I don't know how you would do that, but so going from let me look at the question here. I just had it up.
Sure. They're basically asking how changing from a lab-based adherent protocol to suspension scalable protocol improved the cell therapy and improved the product.
Well, not necessarily. It depends on that suspension protocol. I've been involved in moving towards from an adherent to a suspension lentiviral process, where initially that suspension process had a lot of empty capsids, right? So it wasn't as the LVV wasn't as good, right? And then you define that. I think the real benefit there is in throughput. You get a lot from moving from adherent to a suspension. You have a lot more product that you can make, but also a lot less risk. You have fewer manipulations. You're dealing with cell stacks. You could have a large LVV batch, and it could be 25 cell factories, right? Or you can go to like an iCELLis for large scale, but it's clunky and a lot of manipulations, and every manipulation can impact sterility, et cetera, and activity and things like that. You go to a stirred tank, like a disposable stirred tank type suspension. I think the fact that you have fewer manipulations is actually better. John, do you have any comment on this?
I mean, I largely agree with your experience, Michael. I know in my lifetime, what we've tried to do, I think, going from adherent to suspension-based cultures, whether that be in the world of drug substance for lentivirus or even drug products for cell therapy, in the world of manufacturing, the primary benefit is not, I think, the way that the question was termed. It doesn't necessarily improve the cell product, but instead what it's doing is it's providing an economy of scale, higher efficiency, more throughput, in such a way that you're making the process better and bringing down the COGS. And at the end of the day, that's still the intent is not necessarily make a more potent or a better cell product, but to make the same product but cheaper, such that you can get those
Let's
drug
remember
product
also,
into more patients.
What is comparability, right? We never actually said it. Comparability, from what the regulators have told me, is, and it's documented in the guidance, is demonstrating that process product A is substantially similar to product process B. That doesn't mean it's better. Better could mean a different product.
That's right. Yeah, completely agree. And it's the guidance is very, I think, predicated on efficacy and safety, as we outlined earlier. One of the things we didn't talk about was stability, but that is a key parameter as well, that I think a lot of folks lose sight of when they think about comparability. But that stability piece is absolutely key.
Okay, good. I want to get through as many as we can here in the next few minutes. And for the benefit of the audience who didn't submit a question, I'll pose this one to John. Perhaps you could take this one first, or Mike, or whoever wants to handle it first. Can a smaller container with lesser cells, identical cell density and composition, be used for establishing stability for cell therapy products? If so, what are the requirements for generating comparability? Assume an immuno-oncology cell therapy product as the example. And whoever wants to take that can jump in.
I'm trying to find it so that I can ensure that I'm answering the question that was asked. What number is that?
It is number 6. Okay. From Astellas Institute of Regenerative Medicine.
here it is.
It's a loaded question, so my apologies for the delay in response. There's several of them in there.
I've actually come
Michael,
across this a lot.
go ahead, Michael, if you're ready.
So stability is supposed to be reflective of drug product, right? So as you intend to use it. So theoretically, yeah, you could use a smaller vessel, like a sentinel vial, for example. But you have to prove that that sentinel vial, and you have to do this anyway if you're going to use it for lot release testing. You have to prove that that sentinel vial is reflective of that bag. Personally, I would rather have stability done in the final form. And if I were to reduce that, I would just use the same bag but smaller, because there's a little difference between I think a bag and a vial. The other thing that we need to think about is bags sometimes crack on freeze, and that could be impacting your stability. So my preference would not to be use a vial, but I'm not saying you can't use a vial. There's just a lot of additional things you need to worry about that could be ancillary and change your, if you ever before the FDA or European or any regulator, sometimes you get down a rat hole, and I see this as a potential rat hole in an inspection, kind of proving your vial was representative when you could have just done it with a final dosage form.
Okay. We have time for about one more question. So this particular person would like for you to speak to the high-level considerations of expanding or adding a new manufacturing facility for the BLA when that new facility was not part of the pivotal trial. What are your thoughts on that?
Site change. You'd have to, you have to look at the equipment, processes, facility, the people, the training, and the throughput. So we're required, it's in the CAR-T guidance and various, to validate throughput or the number of widgets we can make over a unit time in our facility. So you'd have to have all that validation in place, and then you'd file that as a site change, I would think.
John, any quick comment on that?
A similar response from me. I mean, it is indeed a site change. It's a major change in the eyes of the agency. So you are going to get grilled on that decision. But it's one that, at a CDMO that we have those conversations quite a bit. And a lot of what's discussed are, how we can basically provide continuity of operations, both on a personnel perspective, but also an engineering or facility-based perspective for whatever has been done previously. And so there's a lot of diligence that needs to be done as part of that decision, and quite a bit of questions that need to be answered. But again, I think high level, the big things there are resource in the world of people and training, and then facility fit to your process. Those are the two things that I would advise folks to focus in on first, and then expand from there.
The other thing that we didn't mention, which I think we kind of mentioned but didn't specifically, which is this notion of reference site. So once you have multiple sites, the expectation is you compare to a reference. So your main site, so I guess the question is, what if you change that reference site? So there's a lot of considerations there. Use quality risk management, looking at your control strategy. If you don't have that stuff in place, you will not be able to evaluate what's required to move to the new facility. It would be a PAI type, inspection type requirement. And I can guarantee you, if you go through that without any of that information, you're not going to have a good time. There was one other question in here about how does this relate to CDMO selection, and I think I think that's central. When I look at CDMOs as a client, and John can attest to this because I went and visited his site, data integrity and quality systems are like 80% of what I'm looking at. The other 20% is facility layout and aseptic processing capabilities within the facility. But I think if you go to a CDMO like where John works, or there's several others that have stellar quality systems, that's where I would gravitate towards, because as you grow your product, you're probably not going to be in a CDMO anymore to meet the needs, or maybe you'll have a CDMO and your own facility. You're going to need to maintain all that data. So I think that's the most important consideration for CDMO.